The current protocol is significant because it allows researchers to produce siRNA-loaded nanoparticles and to adequately characterize these nanoparticles to ensure their suitability for administration to animals prior to subsequent studies.
The advantage of this approach is to produce nanoparticles that increase the bioavailability of siRNA and to produce nanoparticles that are neutrally charged and thus suitable for systemic administration.
This technique is impactful because siRNA nanoparticles can be applied to treat a myriad of diseases necessitating systemic administration, including cancer, cardiovascular disease, and autoimmune disorders, among others.
To begin, dissolve polymer in ethanol at a concentration of 33.3 milligrams per milliliter and stir to ensure dissolution. Then use a pipette to transfer polymer solution in 10 millimolar citric acid buffer to reach a concentration of 3.33 milligrams per milliliter. Add distilled water into a tube containing small interfering RNA to achieve a concentration of 50 micromolar. Mix the polymer and small interfering RNA solutions thoroughly in a tube by pipetting up and down to achieve a nitrogen to phosphate ratio of 10. Place the tube at ambient temperature for 30 minutes. Then, add 1 milliliter of 10 millimolar phosphate buffer at pH 8 to the tube to achieve a concentration of 0.28 milligrams per milliliter and mix gently either by pipetting or inverting the tube. To confirm that the final pH is neutral around 7.2 to 7.5, pipette 10 microliters of the small interfering RNA nanoparticle solution onto the pH test strips.